Hello iGEMers from around the world. We are Montgomery Cougars NJUSA, or more commonly known as Montgomery iGEM. We are a motivated group of high school students from Skillman, New Jersey, focused on innovative solutions to everyday problems. Throughout the year, we have collectively learned a lot from this program. As a first-year team, we are excited to be represented at this competition.
Hi, my name is Susan Liu. I'm a junior at Montgomery High School. I'm the president and founder of Montgomery iGEM.
Hi, my name is Robert Dembinski, and I'm the vice president of Montgomery iGEM.
Hi everyone, my name is Cindy Fan and I'm a junior at Montgomery High School. I'm also the operations team leader for Montgomery iGEM.
Hi, my name is Vivian Hu and I'm a junior at Montgomery High School and I'm the leader of the R&D team.
Hi, my name is Lucy Sang and I'm in 11th grade and I'm the recorder at Montgomery iGEM.
Hi, my name is Sarah Oh. I'm a junior at Montgomery High School and I'm the librarian of iGEM.
Hi, my name is Emily and I'm a junior at Montgomery High School and I am the secretary of iGEM.
Hi, my name is Casey Chow and I'm a junior at Montgomery High School. I am the systems manager for Montgomery iGEM.
Hi, I'm Avi Pula, and I'm a junior at Montgomery High School, and I'm the community liaison.
Hi, my name is Ankit Shah. I'm a sophomore at Montgomery High School, and I'm one of 2 underclassmen representatives in iGEM.
Hi, I'm Joanna Lee. I'm a sophomore at Montgomery High School, and I am one of 2 underclassmen representatives of Montgomery iGEM.
This year, we have been working on developing our team along with our health and medicine track project, Acne Reduction and Inhibition.
We have developed a biological mechanism that will aid adolescents and adults alike in reducing acne production.
Many teens use over-the-counter medications such as Proactiv and Epiduo to reduce blemishes from forming on their face. The most commonly used acne remedies contain benzoyl peroxide, which kills the bacteria that contributes to the inflammation of acne. Although it is often lauded for successfully eliminating the bacteria, the actual chemical can do a lot of damage to our vulnerable skin. Some common side effects include dry skin that flakes and peels, or itchy skin that leaves a burning sensation. This inconvenience occurs because the benzoyl peroxide does not isolate its target to acne. It strips skin of its natural oils and thus cracks the skin. Taking this problem into consideration, Montgomery iGEM sought out a biological approach to attack acne rather than directly eradicating the bacteria, which strips our skin of necessities. Instead of targeting the bacteria, we tried to eliminate their food source, sebum. By creating a treatment that breaks down lipids that comprise of sebum, the bacteria do not have the resources to grow, multiply, and produce the inflammatory response that we all know as acne.
Our inspiration for this project stemmed from something close to all of our hearts, ourselves. Growing up as young adults with acne, we've had to deal with several problems that are a result of this condition. Thus, when Pankti introduced the idea of a mechanism to reduce acne during one of our brainstorming meetings, the board immediately supported her proposition. Though the mechanism was tinkered with multiple times, our goal has always been clear: to find a better way for teens to combat acne.
Acne vulgaris, commonly known as just acne, pimples, or zits, is the most widespread dermatological disease in America and the world. Over 85% of Americans have or have had acne throughout their lives. Unfortunately, over 40% of acne is serious enough to require prescription medicine and leave permanent scarring. This widespread disease is caused by many reasons and is directly correlated with high stress and hormone levels. Acne can manifest itself in either inflammatory or non-inflammatory forms. Common forms include pimples, papules, pustules, cysts, nodules, and comedones.
Scientists have also confirmed that localized vitamin A deficiency can lead to hyperkeratosis, which is critical in the development of sebaceous oils. Sebum is the collective term describing the oils and lipids that the epidermis secretes. When hormone levels, especially androgen levels, are high, more sebum is produced, thereby creating more acne. As you can see on the right, the sebaceous gland is located within the epidermis under the hair shaft. Overstimulation of this gland creates an excess of sebum that results in many forms of acne.
Acne has a few main causes. The first is comedogenic. The earliest form of all acne comes from the microcomedones that form under the outer epidermis layer. While we are not sure of the exact mechanism in which this forms, there is research that supports that these comedones form as a direct result of hyperkeratosis. Hyperkeratosis is the proliferation of ductal keratinocytes that secrete oils around hair follicles. The buildup of oils around the hair follicle can create either open or closed comedones, which we see as white or blackheads. This process is shown through the picture on the left. As you can see, the oils build up and deform the hair follicle.
Alone, comitogenesis cannot create extreme cases of acne. Instead, the blockage of hair follicles is a necessary precursor to P. acne proliferation. On the left, you see a picture of Propionibacterium acne, the bacteria responsible for the most serious cases of acne. This bacteria thrives on the lipids and fats present in sebum. Its inherent lipases create the backbones for acne synthesis. In addition to this, P. acnes also elicit an inflammatory response from the immune system. Phagocytes attack the bacteria and leave the skin irritated.
So, in short, the mechanism for acne occurs in 3 simple steps. First, the sebaceous glands produce or overproduce sebum. Sebum is primarily composed of wax esters, triglycerides, and squalene. The sebum then acts as food for the bacteria to feed off of. Finally, the survival and spread of the bacteria causes the immunoresponse and irritates the skin. In order to reduce acne or inhibit its formation, we knew we had to stop one of these 3 things.
After we had decided on our project, we spent many of our meeting days researching the topic. Our team had a collective brainstorming session about how we would create the mechanism.
Once we had a couple ideas going, we read up on scientific articles pertaining to the pathogenesis, causes, effects, and medications for acne. As you will see, we had lots of difficulty finding a realistic and solid mechanism that would accurately target acne production.
Our first approach to diminishing the nutrient source of P. acnes is 5-alpha reductase inhibition. We found that an enzyme called 5-alpha reductase is responsible for the breakdown of testosterone into dihydrotestosterone. This new molecule catalyzes the creation of sebaceous contents. So, we figured that if we inhibited this enzyme, we could reduce the acne that the breakdown would create. However, this mechanism was not satisfactory because in the real world, only men with severe acne would have appreciable quantities of 5-alpha reductase that we could target. Thus, we deemed that this was not a method with a wide enough spectrum.
Our second mechanism was the complete opposite of our first method with respect to spectrum. This method tried to cater to the most amount of chemicals present in sebum. We tried to devise this mechanism by means of a concoction. Since our fundamental goal was to break down sebum before the bacteria could thrive off of it, we looked for various enzymes that could break down triglycerides, squalene, and other lipids. We found the PNPLA2 gene that codes for adipose triglyceride lipase, the LIPC gene that codes for trisoglycerol lipase, the SQLE gene that codes for the breakdown of squalene, and the LPL gene, which codes for a lipoprotein lipase.
However, while this approach could have been extremely effective in inhibiting the production of acne, it was much too ambitious and unrealistic. One of the biggest problems with this approach was the fact that none of these genes, or even related genes, could be found on the registry. When looking for external sources of genes, we ran into the problem of inconsistent restriction sites, financial hardship, and unavailability. So with this in mind, we decided that we needed a more modest approach to our project. A happy medium between these two, if you will.
Our third idea came from the naturally occurring serum albumin protein. This is the most plentiful transport protein within mammalian circulatory systems. In the human, it is critical for transporting both human and fatty acids. Using this logic, we thought that increasing albumin levels in the facial areas would facilitate more transport of fatty acids away from acne sites. We had even found the ALB gene, and were ready to use this as our final method until we made some serious considerations. The downside to this lies in the fact that it is extremely unpredictable and can do harm when unregulated. We ultimately decided that these risks were much too high and the benefits may not be appreciable.
Our fourth idea revolved around the vitamin A deficiency at the root of comitogenesis. Since hyperkeratosis is suspected to be caused by vitamin A deficiency, our plan was to create bacteria that could synthesize vitamin A. However, upon further research, we discovered that vitamin A is not a molecule native to humans. It must instead be ingested from other sources such as carrots, which contains the beta-carotene needed for vitamin A synthesis. Because of our unfamiliarity with genes from plants and its unavailability from the registry, we decided that we needed to find another method.
Our final solution came from beta oxidation facilitation. Beta oxidation is a natural function of triglycerides and other lipids within the skin that contribute to acne synthesis. By breaking down components of sebum, such as waxes, we can eliminate P. acnese's nutrient source. We found out that most of the triglycerides and fatty acids inside sebum were medium-chain molecules, molecules that contain 10 to 25 carbons. With this in consideration, we first looked at an acyl-CoA dehydrogenase meant to target triglycerides between 10 and 16 carbons long. We knew that this could have worked, but could not find a standardized part in the registry.
Perhaps some luck was on our side and we managed to find both a medium-chain alcohol and aldehyde dehydrogenase within the biosynthesis category of the coding regions. We had long last found our solution to our project. However, upon closer analysis, sapinic acid, a fatty acid associated with human acne, lacked both alcohol and aldehyde functional groups, which is probably why it was problematic in the first place. Just as we were ready to scrap this idea as well, we found that wax esters were rich in alcohol groups, and this mechanism would indeed target and dehydrogenate them.
The following are two separate constructs depicting the mechanism of action. The first consists of a LAC-L-regulated promoter, BBA-ROO10, the ribosome binding site, a medium-chain aldehyde dehydrogenase, BBA-K398005, a green fluorescent protein, or GFP, and the terminator, BBA-BOO15. The second construct simply replaces the aldehyde dehydrogenase with a medium-chain alcohol dehydrogenase. We reasoned that both the aldehyde and alcohol serve to alter the formation of the lipids, rendering the lipids unable to aid P. acnes bacterial proliferation as a nutrient source.
In order to complete this lab, we used a great deal of scientific equipment and solutions. First, we started off with the 4 BioBrick agar stabs that we received from the iGEM registry. This was our first major setback. With no experience of how the registry worked, it took 12 days between our order time and time of arrival. We also used the high school enzyme kit provided by New England Biolabs. Finally, other biological materials include the PSB1C3 linearized plasmid and the promoter and terminator as found in the DNA distribution kit. Aside from biological equipment, we also used many laboratory machines, plastic and glassware, including the thermocycler, incubator, centrifuge, micropipettes, beakers, and more.
Our first step was to grow the bacteria so that we could harvest more of the plasmid, and thus parts within them. We actually did this step twice, further exacerbating our time crunch. This procedure was carried out in 2 distinct steps, growing the cultures on agar plates and then transferring the samples into culture tubes with a 16-hour incubation period between them. The first time we performed this, we religiously followed the 3A assembly instructions and plated the samples out onto ampicillin kanamycin plates. The next day, we had discovered no growth and realized that the new parts were not antibiotic resistant and thus did not grow. We repeated on LB agar plates and harvested half into the culture tubes as the protocol suggests, but inoculated more LB broth so that we could immediately start the mini prep lysing.
Our mini prep was done generally the way iGEM registry's protocol calls for. However, we did not use the 5 ml culture tubes for 2 distinct reasons. We could not spare the day that it took for the bacteria to grow, and our centrifuge could not accommodate the large tubes. Thus, we used scaled-down solutions as well as freshly inoculated LB broth. We solved that problem of small DNA volume through use of distilled water and extremely careful pipetting. At the end of this step, we also rehydrated the remaining 2 DNA parts found on the kit plates.
Our restriction digestion took more cognitive prowess and creative thinking to complete. Since we were going to ligate more than 2 parts to a linearized plasmid, we had to make sure that we digested the DNA segments correctly so that they would not be susceptible to flipping and the likes. We followed the procedure for prepping the solution, but digested the 6 samples of DNA we had with different enzymes than the ones listed. When we had finished, we had all of the parts digested and ready for ligation.
Our ligation process was the most unique part of our experimental procedure. R&D member Yuan He realized that if we simply ligated all 6 parts of the plasmid together in one ligation, the chances of the correct combination of materials would be very slim. Therefore, she developed a method in which we did the ligation in parts instead of all at once. We first ligated the promoter and RBS together, and then the open reading frame and GFP and terminator together. Finally, we ligated all 3 together. We realized one flaw in this plan, in that even if the ORF never made it into the plasmid, the GFP signaling protein could still be active.
Transformation was the final step in our lab process and the most rushed. Because we were approaching our last day of school as well as the iGEM deadlines, we could not perform many parts of this procedure. We did not use a positive control, antibiotic plates, or any inducer, but otherwise followed the procedure for heat shocking, inoculating, and finally plating. We left the lab hopeful of our results.
checked on our bacteria cultures, we found that the bacteria did indeed grow, but did not display the correct reaction when viewed under a UV light. We can only conclude that our bacteria did not accept the plasmid, that the plasmid was ligated incorrectly, or that the promoter was not stimulated. Our suspicion is that our promoter was not stimulated. We recognize that the LACAL-regulated promoter would not be initiated by the LB agar plate. However, due to our limited time, we could not obtain what was needed for our project. Also, during our transformation process, we had to forego the RFP control in order to finish the experiment in time. Our ligation procedure was much more complex than the procedure given, so we had to experiment and create our own ligation process. All in all, our first year laboratory was very shaky and our lack of experience forced us to take many risks. We hope for better results in the future.
So that concludes the scientific portion of our presentation. Since we were a first-year team, our research and laboratory work was quite shaky and we encountered many problems. However, we feel that we have found some solid ground and are excited for the coming years.
Our team is composed of around 35 members of dedicated, inspired, and motivated MHS students in their freshman to senior years. Ever since the beginning of the club, we have worked hard and worked together to build up both our team structure and participate in research and labs.
Our mission statement is as follows. Montgomery iGEM's fundamental goal is to inspire the next generation of synthetic biologists, doctors, biomedical engineers, and STEM professionals through immersion in a challenging and innovative program that allows students of MHS to explore concepts of applied biology.
We seek to spread this message in everything we do, whether it is advocating within our community, working on research for our project, or just during team meeting bonding time. Throughout this year, we have established ourselves as a stable and organized club, ready to inspire the future generations of STEM professionals.
So as a first-year team, we would like to share some experiences and challenges we faced so that future rookie teams can encounter less roadblocks in their endeavors. Our first challenge was our team structure. Since we started in the end of October, we had very little time to pull together a team before registration and the competitive research season.
Looking back, there are many things we could have improved upon, such as underclassmen participation, teaching methods, team bonding, and planning ahead. While we did have regular meetings, some were unfocused since even the leadership was figuring everything out. Next year, we plan on restructuring the team and board so that meetings run in an organized and efficient manner.
Another challenge we faced was research for our project. It took us many weeks to decide on a project that was both feasible, and it took us even longer to devise an appropriate method of attack, so to speak. We were quite confused on exactly what synthetic biology consisted of and spent many hours researching the basics. We found that the first step to successful research was for everyone to completely understand the basis of the problem. Once we had a thorough collective session on the pathogenesis of acne, we were able to devise our final solution.
Our biggest challenge was definitely the lab. Due to academic time constraints, as most of the board is comprised of juniors, we were not able to seriously start until after AP testing. In retrospect, it would have been prudent to fully understand the methods of transformation, ligation, and restriction digestion prior to this. We used the PGLO lab and 3A assembly as practice, but had complications with our final project.
As our team continues to grow, we have high hopes and aims for future years. One of the biggest things that we hope to begin is community outreach. iGEM is relatively new, and we would like to spread our message through presentations in our district and beyond. We are also planning on coming up with a comprehensive future plan for the team and structure over the summer, so that we can start off next year more ready for our project. Through this, we hope that we will spread our passion and inspire others to pursue careers in STEM, especially careers involving applied biology. Finally, for the future, we hope that our research and project will be more successful. While we have made a great start, we have a great deal of improvement to go.
The team would not have made it this far without the help of our mentors, advisors, and instructors. We would like to thank the MHS administration for being so supportive of our endeavors, and especially the science department for staying the long hours and teaching us what we needed to know. We would also like to thank our advisor, Mr. Christopher Resch, for being a phenomenal teacher and supporting this team in times of hardship. The things he has taught us have inspired us to work harder in the future and achieve our high goals.
We would also like to thank Mr. Jason Sullivan, our science department supervisor, who has helped us in innumerable ways. Additionally, we thank our college advisor, Gavin Kuziel, for his contributions and initial inspiration to start the team. Finally, we would like to recognize our outstanding R&D members, Neha Jugani, Yuan He, and Pankti Kothari, and our photographers, Shannon Liu and Joanna Lee. It's been a wonderful year, and we thank the iGEM organization for this amazing experience.